Hi guys, welcome back. We're going to go into a new topic, and this is going to go into that different organelle. This is going to go into the nucleus. Now remember, the nucleus's job was to hold DNA, and then remember the DNA's job was to actually hold the instructions to which protein to make. Or if we're talking about the cookie analogy, it was the nucleus was like the office and the DNA was like the recipe. So we're really going to get into just DNA structure, what it looks like, and then how it replicates itself. So this is DNA. We see that it's nice and coiled, and it's really, really long. If you look at the DNA poster over there on the right, you will see that there's like millions and millions of pieces of DNA just to one chromosome, and we have 46 chromosomes. So DNA is a biochemical known as deoxyribose nucleic acid. So once again, because it's a nucleic acid, we have a monomer that makes it up. And DNA is what makes up chromatin or chromosomes. So remember the difference when we talked about meiosis and mitosis. Chromosomes are that typical X formation, or if they're just in the cell by themselves, we call them chromosomes. Chromatin is when the DNA is like this. It's uncoiled, it's relaxed, it's stringly. Don't get too caught up between chromatin versus chromosomes, because when it comes down to it, they're both made of DNA, and they both control which proteins are made. Here's a good picture. You have chromatin when it's relaxed, and then it's going to actually go and bond to these histones and wrap around it and coil really tight, and then we actually make the condensed chromosome. Remember, we have 46 of these. So as a review, chromatins become chromosomes, and then it's a section of these chromosomes that determines a trait, and that is called a gene. So I wish I was there to kind of show it to you, but like, here's a chromosome. So from here to here is going to be a gene. It's going to have different sequences, A's, T's, C's, G's. It might be 4 letters long. It might be 400,000 letters long. But this section of DNA will make a protein, which therefore makes your trait. So a gene is what determines your trait. I'm going to say that again. A gene, which is part of your DNA, determines your trait. Nothing big here. It's the blueprint for proteins. It's the organism's traits. So this makes sense. You look like your mom and you look like your dad. It's because you got half of them from mom, half of them from dad. Part of your DNA is known as genes, and it determines your traits. All right, so where do we discover this? These two guys are the ones who get credit for discovering, Watson and Crick. And they're the one that discovered, or have the credit of discovering, the shape of DNA. And they termed it the double helix, and this is what that twisted ladder appearance it is. Now, I appreciate Watson and Crick. They found this in the 50s, and they were kind of odd guys. They look odd even. But they were extremely brilliant, and they were trying to figure out how these chemicals would actually come together, condense so tightly, and fit in the nucleus. Now, the reason why it's called a double helix is because it resembles a twisted ladder, just like that staircase you go up. You're going to have rungs on the side, and then you'll have like the rungs of the ladder that you walk up to. However, they couldn't have done what they did without this woman named Rosalind Franklin. So Rosalind Franklin, this was in the 50s. She worked at King's College, which is in England. And she was a woman, and the woman in the 50s did not get much credibility. She was actually an X-ray topographer. She was into discovering different types of X-rays. Well, she worked under a guy who was kind of a jerk, who was friends with Watson and Crick. Well, Rosalind Franklin took pictures of DNA, had hard concrete evidence, did this multiple times. This is actually the picture she took. It's actually called Photo 51. And this proves that DNA is a double helix. I know, it's hard for us to determine, but it's an interesting story. So Rosalind Franklin got frustrated by working underneath that guy, so she left King's College. And by chance, somehow, during the move, She left her research and her papers on a desk before they could get packed. So by the time she went to a different university, the guy she worked under actually took her research, discovered this picture, and took it to James Watson and Crick. Guess what they did? They looked at it, said, yes, this is exactly what we need to prove that DNA has a double helix, and they published it. And they ended up getting the Nobel Peace Prize for it. Rosalind Franklin never received credit for it. She actually died in the early 60s from ovarian cancer, probably from the x-rays. But now in the last 10 to 20 years, she's starting to get credit and fame for it. Anywho, interesting story. Next. So this right here is DNA. You can see that it's twisted ladder. I've ironed it out. So you have the sides of the ladder here, 1 and then 2, and then you have the rungs that walk up it, and it's just twisted in addition to that. So it's a polymer that's made up of these monomers known as nucleotides. This is all a review for you. Nucleotides are made up of deoxyribose sugar, phosphate, and nitrogen base. We will get into the other type of polymer known as RNA tomorrow. But today we're just doing DNA, which is sugar, known as deoxyribose sugar, phosphate, and a base. So in this picture, phosphate is the P, sugar is right here, and then this is going to be your base. Notice my backbone is a sugar-phosphate combination. Phosphate, sugar, phosphate, sugar, phosphate, sugar. These are your different bases. And this right here is really what's going to determine what our protein looks like. The nitrogen bases are going to be divided into 4. We have adenine, represented by the letter A, guanine with G, cytosine, C, thymine, T. So these are the only 4 that DNA has: adenine, guanine, cytosine, thymine. And we say these bases are complementary, meaning they are complementary to each other. Meaning adenine is going to only bond with another one known as thymine. Guanine is going to bind with cytosine. So let's see what I mean by that. So one side of the DNA will only bond to the other side based upon its complementary pairing. So we know here's our sugar phosphate group. If this is our base, which is known as cytosine, we know the other side of DNA is going to have to be guanine. And then there's the sugar phosphate base. So adenine bonds with thymine, guanine binds with cytosine. I like to remember apples in the trees, cars in the garage. A to T, C to G. Apples in the trees, cars to the garage. A to T, C to G. So pretty much if I tell you this side, one side, you will be able to tell me the other side. So if I have adenine on one, you or know the other side is going to be thymine. This is the central dogma of biology. This is fascinating. Every single living organism that has DNA, which we know is a characteristic of life, contains the exact same 4 nucleotides: adenine, guanine, thymine, and cytosine. But it's the combination of these that makes it so different. And the way I like to kind of explain it, let's say we're looking at me who has brown hair. So in my DNA, I have the gene for brown hair. But my letter combination might be A, C, G, T. And then let's look at someone who has red hair. Well, they have the gene for hair color because they have hair color, but their combination might be ACCTA. So they still have the same nucleotides. However, they have a different combination or different series. And that's what makes it different for everybody. So it's kind of like this. Here's gene 1, which might be for hair color, and mine looks this way. Someone else who also has a different color hair still has gene 1, but they have a different combination. Central dogma. So here's a good example. Here's cytosine. On this side, we know it bonds with guanine, and of course we are going to practice this. So this is important in helping us realize what DNA replication does. Now let's think back. Do you remember when DNA replication occurs? Give you a second there. It's a process we've already learned. It's going to be in mitosis and meiosis, specifically in which phase? Interphase. Okay, so let's go over DNA replication. So anytime you are going to replicate, you are going to make a copy. Why? Because you need more additional DNA into the next cell. Let's see, sorry, time for that. Now, in order to replicate it, an organized process has to take place. And it's actually a really simple process because our bases are complementary. If A is over here, T is over here. So really what we have to do is unwind it, as you see here, and expose those bases. So if we know one is A, we know we need to bond a T. So let's get into some more details with this. So first we have to have this enzyme known as helicase. A, we know it's an enzyme because it ends in ASE. And this enzyme washes with the DNA and it unwinds the helix. It's kind of like the ironing board of the DNA. Then we have an enzyme that breaks apart the rungs of the ladder. It breaks apart the A and the T's, or the C's and the G's. So it opens it up, leaving like the sides of the ladders exposed. Then we are going to free-floating nucleotide, will actually bond to the open bases. So each original strand will now have one new and one old. And then eventually you will have two of the same identical strands together. And then you have two pieces of DNA. There's a replication video there, Mr. Williams. I think Ms. Hartman showed you to play it. It really helps here. So let's kind of look at this. Great picture here. So here's our DNA, double helix. We have enzymes that flatten it out, just like this, and then open it up. So we're exposing two different sides. So right here, I'm going to start back here with G. Guanine, I would attach a free-floating cytosine to it. Cytosine on this side, I would attach a free-floating guanine. And I would just build upon it as I go. So my end result, I have two strands, one and then two. And look, they are exact copies. GC, GC. One strand has one old piece and one new piece. The other strand has one new piece and one old piece. They are identical to each other. All right, so now what does this mean? What do you have to do with this information? We're going to practice it. So this is a DNA strand. So what I have done is I have taken, let's see, this strand right here, and I have flipped it on its side, and I have grouped them in groups of 3, which are called codons, which we'll get to later. So what I would do is I would give you a piece of DNA, and I would say either replicate it or give me the complementary strand. Both are asking the same thing. When we think replicating, we would be like AATC. No. You're giving me the complementary base strands. So for example, let's start here. Adenine would bond with thymine. Over here, thymine would bond with adenine. So I'm going to give you a couple of seconds to do that. And Mr. Williams, you might want to pause to give him time to do that. And if you finished it out, this is what it should look like. A with T, T with A, C with G, G with C, T with A. So let's practice another strand. Write this down on your paper. You need practice. And Mr. Williams, you can pause because I'm running out of time. All right, and then here's the answer to this strand. And then here's the next one. So pause again. Write it down. And then look. G, 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 C, C, T, A, C, C, T, A, T, G, T. Oh, Miss Finnison, you made a mistake. Cytosine goes with guanine, but look what went in here. Thymine. Dun, dun, dun. This is known as a mutation, and we will get into mutations later. All right, so you got an activity. DNA lives in the nucleus, complementary strands, adenine with thymine, guanine with cytosine. You give one side to the other. The reason why you replicate, so you have two copies. I'm running out of time. Okay, bye. Have fun practicing. Talk to you later.